Hello friends, welcome you in Research Needs. In this video, we will see the commonly used instrument in experimental pharmacology. So we are going to see this by using Expharm software. So we will go and see that. So this type of interface you can see after clicking here. You will see the list of instrument. First, rotarod, ectophotometer, tail flick, histamine chamber, hot plate, elevated plus maze, electroconvulsiometer, pole climbing, plethysmometer, metabolic cage, organ bath and chymograph drum assembly. One by one we will see the all instrument and listen the description about those instruments. So we will see here.
Rotarod apparatus. Rotarod apparatus has a horizontal grooved rod rotating at a fixed speed. The mice are made to balance on this rod. Dependent upon their motor coordination, central nervous activity, and grip strength, the animals either stay on the rotating rod for a specific time and after that fall down on the platform of each compartment. The floor of each compartment has sensors that deactivate the timers, and the exact fall off time for each rat is displayed on the respective display. Applications: reduction of motor coordination, CNS depression and skeletal muscle relaxation can be studied with the help of this apparatus. Actophotometer. Actophotometer has a central chamber with an arrangement of light sources and photocells at the base of two opposite walls. The light of each source is focused on a photocell. Any interruption in the path of light activates the photocells, and this is counted as a measure of horizontal locomotor activity of the mice kept in the chamber. Applications: the locomotor activity can be determined actophotometer to study CNS depressants stimulants. A tail flick apparatus. The tail flick apparatus is used in the test of the pain response in animals, similar to the hot plate test. It is used in basic pain research and to measure the effectiveness of analgesics by observing the reaction to heat. It was first described by Dummer and Smith in 1941. Applications: a study of the analgesic activity of any drug can be done by using this apparatus. The analgesic effect can be estimated in animal models of analgesia through this apparatus. The time taken by the animal to reveal pain sensation to escape the painful stimulus, withdrawal of the body part on which pain is inflicted or tail, is determined. Histamine chamber. A histamine chamber is a lab equipment used to examine how histamine or other compounds affect animals, particularly guinea pigs. It is usually constructed of clear plastic or perspex. It has a nebulizer built into it to release aerosolized materials, and the chamber allows researchers to observe the animal's response to histamine. Applications: histamine chamber is basically used to evaluate activity of antihistaminic drugs on animals because antihistaminic drugs inhibit the inflammatory effects of histamine on drug-treated animals. Hot plate apparatus. Eddy's hot plate is a device used to give heat stimulus to the paws of animal. A metal plate at the top of hot plate apparatus can be heated in the controlled manner and maintained at the required temperature for the duration of an experiment. Generally, in testing of the analgesic activity of drug in animal, 55 degrees Celsius temperature is used as a thermal stimulus. Applications: study of analgesic activity can be done with the help of hot plate apparatus. The effect of analgesic drugs can be evaluated in animal models of analgesia. The time taken by the animal to reveal pain sensation, withdrawal of the body part on which pain is inflicted or tail, is determined with the help of this apparatus. Elevated plus maze. The EPM, elevated plus maze, apparatus consists of a plus-shaped maze elevated above the floor with two oppositely positioned closed aims, two oppositely positioned open arms, and a center area. The EPM is designed to utilize rodents' inherent dislike for open and elevated areas, enabling researchers to examine the impact of anxiolytic, anti-anxiety, and anxiogenic, anxiety-inducing medications or other interventions on their anxiety levels. Applications: measuring anxiety, screening for anxiolytics and anxiogenics, understanding anxiety mechanism studying, the effects of treatments. Electroconvulsiometer. Electroconvulsiometer is used to deliver the electric shock of required intensity to the subject for required duration. In this apparatus, an electrical stimulus with an intensity that induced characteristic convulsion is applied to the animals through the electrode placed on the ear pinna. Further, the duration of tonic and clonic seizures is measured. The drug to be tested is administered to separate group of animals, and its effect on such duration of such convulsions is measured. Anticonvulsant pharmacological agents reduce the duration of seizures induced by electrical shocks. Applications: this instrument is used to evaluate the anticonvulsant effect of pharmacological agents against electroshock-induced convulsions in experimental animals. Pole climbing apparatus. Pole climbing apparatus has a grid floor that acts as a source of shock. In the center of the roof there is a wooden pole. The apparatus also contains one buzzer. Applications: evaluation of sedative and antipsychotic drugs can be done by using Cook's pole climbing apparatus. Animals treated with sedative hypnotic drugs do not show condition avoidance response, response to buzzer do not climb to pole, and also unconditioned response, response to mild current, do not climb to pole, in Cook's pole climbing apparatus. Animals treated with antipsychotic drugs do not show a condition avoidance response, response to buzzer do not climb to pole. Plethysmometer. The main purpose of a plethysmometer is to measure changes in volume, especially in rodents, in order to evaluate inflammatory or edema responses. It's a useful instrument for evaluating possible anti-inflammatory or anti-edema compounds. The device measures variations in pore volume, which may be a sign of swelling or inflammation. Applications: anti-inflammatory medication screening. Researchers can evaluate a drug's ability to reduce inflammation or edema by assessing changes in pore volume after inducing an inflammatory response in rodents. Assessing edema. The degree of swelling in a pore or other body part can be measured using plethysmometers. Tracking the efficacy of treatment. Researchers can determine how well a medication is lowering swelling or inflammation by monitoring changes in pore volume over time. Metabolic cage. The metabolic cage is designed to allow measurement of fluid intake and to separate and collect feces and urine for numerous qualitative and quantitative determinations. In addition, the metabolic cage permits observation of the animal, feces, and urine at all times. Applications: urine and fecal collection. Metabolic cages are designed with a grid floor that allows feces and urine to fall through into separate collection tubes, ensuring minimal contamination between samples. Nutrient studies. By collecting samples, the efficiency of nutrient digestion, absorption, and excretion can be determined. Pharmacological studies. Studying drug metabolism by analyzing the excretion of drugs and their metabolites in urine and feces. Toxicology. to examine the effects of toxic substances on an animal's metabolism by analyzing urine and fecal samples. Behavioral studies, to study the relationship between an animal's behavior, such as food and water intake, and its metabolic processes. Organ bath kymograph drum assembly setup. An organ bath and kymograph drum setup is used in experimental pharmacology to examine how medicines affect isolated tissues. The kymograph records the tissue's response, such as contractions or relaxations, while the organ bath gives the organ a regulated environment. Organ bath has a chamber that replicates the natural environment of the organ by filling it with a physiological salt solution, such as Tyrode's or Ringer's solution. A thermostat to control the temperature. A gassing mechanism to regulate the solution's pH. A hook or clamp, for example, to which the organ can be attached. Kymograph has a revolving drum, usually composed of stainless steel. A pen or stylus fastened to the drum. A recording lever that sends the movement of the organ to the stylus.